we've been doing a lot of work in quantitative genetics the last couple decades, but you also will start to see in this talk that we are really shifting a lot of the focus of what we're trying to do to use those evolutionary tools and population genetics and quantitative genetics tools to really tackle some of agriculture's contributions to greenhouse gas emissions. And so I'll kind of walk you through how we think how evolution and PopGen and so on can play a really key role in tackling some of these issues. And so I'll start out with that talk here. So yeah, so let's get started here. And so the kind of the question, that we've been thinking about and struggling with is we know there's a bunch of species out there that are related to our crops, and frequently we see them on herbarium sheet like this. And this is how we, pretty much the only way we can get access to them. But what we want to do is be able to predict what are their genes doing, and from that, is that making them more adapted to cold temperatures or hot temperatures or some other type of environmental adaptation. And, so this is really kind of the fundamental question we're trying to ask now is how do we tap a globe's worth of evolutionary data to allow us to make progress in some of these goals I was mentioning on greenhouse gas emissions. And so to acknowledge, the funding for this research, most of it's come from the U.S. Department of Agriculture and the National Science Foundation. But a lot of the goals we've been interested in have come out of ARPA-E, the Gates Foundation, and USAID. And so when we think about the crop I focus most of the time on in maize, it and soy are some of the most productive crops in the world. But over the next decades, they're going to be challenged by extreme weather, nitrogen issues, and a real need to increase carbon out of these systems. And so I think a lot of us understand on the climate change side what a lot of this looks like. It's going to be warmer temperatures, extreme weather events and drought and heat waves. And then one things people may not understand quite as much, you get things like wet soils. And also those wet soils, when they're still warm late in the fall or early in the spring, can release tremendous amounts of nitrous oxide. And that is a potent greenhouse gas. The second area, we're in a period now where we absolutely need our plants to capture as much carbon as they possibly can, and be as efficient as they possibly can per unit area. And we've known for several decades how much more productive in terms of fixing carbon perennials can be, such as miscanthus. They can fix 60% more carbon per year than even a really efficient crop like maize. And then finally, the other really big issue out there are these nitrogen problems. Over half of corn's energy input has to do with its fertilizer, and over 80% of its greenhouse gas emissions have to do with nitrogen, either nitrous oxide emissions or nitrogen fertilizer. And so when you look at the greenhouse gas challenge, these are the numbers for the United States. What you'll see is that agriculture contributes about 10% to US greenhouse gas emissions. And agricultural soil management, which is pretty much the nitrous oxide coming off of fields predominantly from those maize soy fields, is by far the biggest component of greenhouse gas emissions, essentially twice as big an effect as, for example, cattle methane releases. And so the question is, how do we tackle that? There also is an opportunity in this space. Maize and soy, if you look at the total biomass at the end of a growing season, how much CO2 is out there, it's equivalent to essentially all the fossil fuels that are burned for electrical generation in a year. And if that same acreage were optimized for maize, soy, and perennial like miscanthus, in 2 or 3 decades from now, we could actually have as much carbon on those fields as essentially is being released by industry and electrical generation. So right now, the crop is producing far more greenhouse gases than it's taking in, but there is an opportunity to really go from being a problem to being a major negative element and contributing to greenhouse gas emissions. And so if you look at what's going on right now, we have this current soy maize market where you have these crops are bringing in tons of CO2 each year. They're releasing lots of nitrous oxide. They support highly valuable markets. And then there's at the end of that respiration, that CO2 goes up into the atmosphere. And so but these markets are likely to be changing. And if you look at the value of these, what CO2 is at anywhere from $50 to $100 per ton, and we're modeling here at $50 per ton. Essentially there's a negative externality of cost of about $17 billion there, and essentially a lost opportunity of capturing the CO2 the value of that carbon that goes through these livestock and ethanol. But I think we're moving into a world where essentially we're really optimizing, you're going to be rethinking about what's grown on this type of land with a mixture of things like perennials like miscanthus, maize, and soy, where we're really are able to tackle this nitrous oxide problem. And then these are the raw carbon compounds that go into a much wider diversified set of things that can't be easily electrified. And essentially, once they go through these types of processes, then there is opportunities for large-scale carbon sequestration. So I think, if we can deal with some of these nitrogen problems and some of these other ones, we can actually make this into a real key tool in tackling greenhouse gas emissions. And so we really need multiple strategies here. It's not just a crop thing. There's all sorts of agronomic issues, from no-till, changing application routes, using cover crops, microbial fixation, and using things like weathered silicates. But one of the strategies that really up till now hasn't been tackled very much is how to actually use the genetics and physiology of the main crop to tackle its greenhouse gas emissions. A lot of the things that surround it have been really the big focus there. And so what our thinking is in this area is, over the last 10,000 years, maize has really been optimized as a feed and food for humans and livestock feed. But maize isn't really that good, especially on the nitrogen side, which is some of what that's all about. So what we're talking about is really optimizing maize for carbon yield and nitrogen conservation. And so I think there's an opportunity to walk up an adaptive landscape without going through a giant dip, because we're essentially working towards its strengths. And so the way we intend to kind of do this is essentially using the 4 key elements that we use all the time in breeding. The first is tapping diversity. And, in that tapping of diversity, where we're looking to try to do is really tap over 1 billion years of evolutionary time to learn how crops recycle their nutrients and fix carbon efficiently. And so, over the last several years, we asked the question, what's the best way to do this? One way was we could look at the evolution of maize and more genomes. And if we did that, we'd essentially tap how did about 3 million years of independent evolutionary time evolve. But what we decided to focus in on was instead try to go sequence 700 genomes with Andropogani. And these are essentially the wild relatives of maize and sorghum. And essentially, they have you count up all those branch lengths, about 1.2 billion years of evolutionary time. And the key thing is they do share this really efficient type of photosynthesis and a very shared type of development. And so, with our colleagues, Toby Kellogg, Matt Huffer, Jeff Rossabara, and Adam Siepel, we're doing this. And one way to think about this experiment is essentially, it's a saturation evolutionary analysis. Every ancestral base has been mutated about 50 times. Constraint can be measured directly off these. And then we're essentially training a series of functional biology and machine learning models to try to figure out how each of these species have adapted to their environments and then leverage that knowledge into maize. And they have adapted to a lot of different environments. Although this clade is only about 18 billion years old, it now occupies and is the dominant species on 18% of the globe's land. And that was before these crops were domesticated. That's kind of the native levels. And essentially there's been convergence for temperature adaptations and rainfall adaptations and all sorts of things over and over again across this clade. Annual to perennialisms have evolved over between 15 and 20 times in this clade. So we're in the process of sequencing these. And so getting these 750 genomes of the Andropogoniae together. And that includes, we've now pulled together 30 high quality assemblies, and then whole genome sequencing from anywhere from 600 to 700. We're seeing how many of them really pass through all the various tests there. And then we're trying to do a detailed analysis of selection at all the genes and their regulation. And so this project got started really with Toby Kellogg's group, but Taylor Aubuchon went around in the middle of COVID collecting samples from all over the country, and from went to Australia, and then in the last few months it's been able to get to various herbaria to collect additional samples. And that really was at the heart of our ability to sample an entire clade's worth of species. But because we have had to use so many herbaria samples, that does mean, you know, we don't have expression data and all sorts of other types of data for many of these species. Matt Hufford's group and Arun have essentially gone through and using long read sequencing technology, been able to assemble a series of 30 different genomes across this tribe. And many of these genomes are as high quality as even the maize genome is today. So essentially, we've been able to create a backbone set of species that we can compare and get a really good idea of what the gene complex is. And many of these are polyploids and other very complex genomes, but still Matt and Arun have been able to pull off some very nice assemblies of these. And so our group is now really asking the question, what traits can we tap out of this clade? You know, the traits that we want to go after in maize are essentially, we want to be able to get it so that it has cold tolerance. It can essentially overwinter. It has the tools for biological nitrification inhibition. It's able to deal with extreme heat. It's able to recycle its phosphorus. It's able to recycle its nitrogen. So we think we're moving into an environment where we don't need the nitrogen and phosphorus in the grain anymore. And instead that can stay on the field just as a perennial would and recycle those. And so the questions were, so we know we've got this diversity and we're now trying to figure out how we tackle that. We also in this area are pushing forward on, you know, how do we use essentially controlled genetics to tap some of this diversity. And I'm not going to go very much into this area, but I would say, and this is really a call out for anybody in engineering and out there, I would say the biggest obstacle to us really making big gains in tackling some of this greenhouse gas and nitrous oxide is we need tools to measure nitrous oxide coming off of plots at a plot scale very cheaply. And that means really being able to measure nitrous oxide release at thousands of locations across the field all year long. And so if anybody has engineering friends out there, this is probably, you know, for those of us in the crop side, we know what the problem is, but this is a key tool we're kind of missing in order to drive the breeding forward. And so the next thing is we know that we can make a lot more progress if we can essentially breed based on markers, either marker-assisted selection or whole genome prediction. And that really has been at the heart of a lot of the revolutions in breeding over the last 20 years, is that essentially genomic prediction allowed this acceleration of breeding that allowed us to make decisions every 4 months to a year, as opposed to a normal process of evaluation that would take up to 5 to 10 years. And so those tools are now available for us to start tapping diversity in a much more rapid way. But the problem we have in these areas with these technologies is right now, those tools work very well within population. And so if we have a big data set from a particular crop, we can make very accurate predictions, but they predict very poorly, sometimes out of population. And if you have a small species training on them, they also can have relatively modest prediction accuracies. And essentially, we're also not leveraging any information from one species to another. And, you know, so I think one of the key questions our group is asking right now is, and others are, how can we add some of what we know from reductionist biology of the central dogma of gene regulation, protein sequence, and physiology to essentially improve these types of models? Because if we get better types of models here, we're also then going to be able to deploy the knowledge and the germplasm that we have, and I talked about at the beginning there, more rapidly. And so we don't think this is going to replace the standard genome-wide predictions, but we're hoping to get improvements in these. And I want to mention, why does genomic selection falter here? Well, some people say, well, genomic selection isn't, or genome-wide prediction isn't mechanistic. But it really is, it's a mechanistically accurate model of inheritance, but it's not a mechanistic model of molecular activity. And really what we want to do is we want to make decisions and tap germplasm using both of these tools. And so finally, we now have also the tools of things like really molecular design, genetic editing, and technologies like CRISPR, that if we can identify those causal variants in whatever species, we now then have the tools to really move them into our target species. And so we don't really see this as necessarily competing with genomic selection and other types of approaches, but really it's just a complement of a new way of adding genetic variation into those pools and making genomic training models selecting and moving much more rapidly. But it allows us to go to more diverse germplasm without disrupting our elite varieties. So those are essentially the tools we have at hand right now, but it still goes back to this question. We know that a lot of the traits we want, whether that's nitrogen recycling, temperature tolerances we're looking for, they're in these species that are related to maize and sorghum, but we can pretty much have them on a herbarium sheet. And so, really we're trying to infer how a dead plant works. And I think, as you'll see in this, we've been focusing in on creating a series of models that use both evolutionary information combined with mechanistic models to try to make some of that inference. And I think, in the last year, the greatest example of using that type of strategy has been AlphaFold 2 and its ability to fold proteins, where essentially they're using a combination of evolutionary knowledge and structure to predict the three-dimensional structure of a wide range of any protein you put in there. Of course, not the whole thing, and not all regions get folded up and are accurately predicted, but it really is a tour de force of science. And so I think that basic strategy, though, is the right one for how we can efficiently go about creating useful models. And so we've been creating models at different points across the central dogma here, from the genome to protein to whole plant. And let's start first talking about some of the modeling of gene regulation to the whole plant. And the first thing we kind of really want to do is say, well, if we could have good models of gene regulation, would that actually help us in doing some of these targets of genome-wide prediction? And that was the question and issue that Andrew Geary and the group asked, is, if we took what our observed expression patterns were, could those help us make more accurate genome-wide predictions? And Anju knew that essentially those expression patterns were the combination of what was going on genetically at the cis regions located very close to the gene, trans-regulating factors, and then various environmental factors in the air. And if she could deconvolute those and just focus on the cis ones, she'd be able to start making more useful predictions, because essentially all those trans factors are cis somewhere farther up the regulatory network. And so what she did is she just used partition the genetic variance of measured expression using a trans effect model that was modeled after the genome-wide haplotype relationship matrix. And the cis effect was modeled on the local cis haplotype relationship, and was able to get a good estimate of the cis haplotype effect for every gene across a wide range of different alleles and tissues. And so what she found was essentially, when she broke those down, is that essentially that cis component of expression explains about a third of expression, and trans explains the other 2/3. So when you just use the observed levels of expression, they're sometimes not the greatest predictor for what you want to do in making causal inferences downstream. But it is a substantial portion, and it was in general quite partitionable between these two.
Hey, Ed.
Yes.
You said it's all cis somewhere further up, right? But are there no protein coding differences in this? Like, it doesn't all have to be expression, right? I mean,
no. So our model here, we're not modeling any type of protein coding here, but those protein coding changes would essentially be in the relationship, the general relationship matrix. So, okay. So yes, they're in there, and essentially that trans component is made up of expression trans and expression protein coding trans. So both of them would be in there. Huh?
If we could really disentangle that and answer that old question, if it's all if it's all regulatory evolution or what, right? I mean,
yeah, no, yeah. So we didn't we tried to disentangle cis versus trans, but we don't know that all of what we saw. What of that trans portion was protein versus cis.
So, I mean, maybe a corollary to that is how stable are those variances, or like how correlated are the cis and trans kinship?
So in those cis and trans, there's a group of genes that have some real outlier. I mean, there's a group that are relatively low heritability, and we don't do a very good job of partitioning them. These ones that are kind of extreme are essentially just presence absence genes. So there's a wide range of variation here. That's the mean, and it's pretty much all over the board there. I would say these tissue ones, I think, are the kind of the interesting thing, that I was excited to see.
Yeah, I mean, I guess I would assume they're highly correlated, the cis and the, I mean, these are like something like a kinship, yeah, or. Well,
the cis relationship matrix is not well correlated with the trans very
No,
well.
It's not, okay.
Yeah,
okay.
No, that's the cis is, that's just one small region of the genome, and it's not particularly correlated with trans.
Okay.
So, yeah, so it's almost, I mean, we played around with incorporating both cis and trans, and actually you can do just a fairly good job just with the cis model, because essentially the trans is almost random there. You extract most of the signal just using the from the cis relationship matrix.
Oh, cool.
And then the neat thing was then Anju then measured this expression correlation between tissues. And she saw between this is just measured expression. I think she sees what a lot of people see. There's some genes that are pretty much zero correlation. Some of these are probably some of those genes we're not scoring and measuring very well, but generally it's positive, and there's a positive correlation between alleles. And then she just looked at the cis component of this, and what you see is there's a really substantial shift towards much more positive correlation between tissues. And so that is suggesting that once you remove the trans component, that cis component is fairly consistent across tissues. So allele rank is consistent across tissues, and we can use that to make genome-wide predictions. And so that's what Anju did relative to baseline of using a haplotype-based approach. She trained on a small set of 280 lines to predict a larger population, and essentially got improvements of 4 to 6% improvement in genome-wide prediction accuracies. And then she did the reverse, where she trained on this NAM population, predicted another one, and got even higher prediction accuracies, up to 14% of her baseline improvement. And I'll give you kind of a feel. Sometimes when we played around with other types of models, just changing various types of genome, we get improvements on the order of a couple percent. So this was actually a fairly substantial improvement to see generally across a wide range of traits to get this level of consistency. So I think that what this suggests is if we can identify and create models to estimate that cis haplotype expression, we could likely improve genome-wide prediction for nearly all traits. Now the question is, how well can we actually predict just from straight DNA sequence that? Because here we're having to essentially impute it. So the other area that we've known has been kind of a key for those cis regulation is essentially deleterious mutations and how they affect expression. And this is an area where Jim Birchler has advocated is really at the basis of hybrid vigor and lots of other traits that balance expression results in a highly vigorous organism. Unbalanced expression results in a less vigorous organism. So these deviations in expression should matter. A couple of years ago, we did models just based on straight expression, and we're able to say, yes, deviations in expression away from that mean, those absolute values, do explain a substantial portion of seed weight. And so where we've gotten to in the last year or so is then, at the time that we knew those deviations existed, we didn't really know where exactly those variants were in the genome. And this is where Baoshan Song has developed a couple new algorithms for really aligning conserved non-coding regions across maize. And so he developed some novel alignment algorithms there. And then that was allowed him to identify with very high resolution these small conserved non-coding sequences. And it turned out that when he started looking at what those were, he got, many of them were highly enriched for transcription factor binding sites and other key open chromatin domains. And so then when he looked at those deviations in expression, the negative deviations in expression were frequently at these CNSs where they were being lost. So low expression really was, we now know nucleotide by nucleotide really what some of those low expression patterns are being caused by. And there are some of these various regulatory elements we can spot by aligning maize versus a wide range of other grasses. And so those are the first are saying is essentially, yes, if we can get that cis component, we can do a better job on genome-wide prediction. And I think we're beginning to start cataloging, base pair by base pair, some of those causal variants that are really key for getting in there. We need to start plugging in these 700 other species, but I think we know now the route that we're going to kind of go to get those causal variants. So the second area we've been trying to work on to get to make inference here is really essentially machine learning models that give us this base level modeling of both transcription and translation. And a couple of years ago, we worked with Silong Zhang's group, and Catherine Mejia and the group essentially then created a series of machine learning models to model over 100 different transcription factors binding sites, and was able to come up with reasonable accuracy for a lot of these models. And I think the really key thing that came out of that study was because of the comprehensiveness of this analysis, they were able to, Catherine was then able to cluster the models together. And that's what the tree on the left is showing, is that's how the models cluster, these 100 and some different models. And what you see is that those models are then colored by their classification in Arabidopsis. And so maize models essentially cluster and fall out in the same way that the known Arabidopsis transcription families fall out. And so this really does suggest that the Angiosperm regulatory grammar is pretty much the same and shared across these species which have 130 million years of evolutionary time. So I think this is saying that we really need to just create one Angiosperm set of models to do a lot of these types of things. Catherine also then created some models to predict open chromatin within maize, and those were quite accurate. And then that got followed up by Travis Reitzman in the group. And he has a paper that's just coming out right now where he essentially trained open chromatin models across 11 different species. And what he saw is that his within species models, their accuracy was roughly the same as the within species models were as accurate as across species models. So again, this is suggesting that in general, we can do a fairly good job or create more universal models across all these angiosperms. So the last area I kind of want to, in this area on the translation side, get into is essentially what are the real key variants for translation. And this is an area that Joe Gage has looked into. This paper will show up in PNAS very soon, but it's essentially, he was looking at protein levels and RNA levels and DNA variants. And he asked where was the variation coming for both rare alleles and common GWAS variants. And for both those sets, they were highly enriched in the 5' UTR. So it wasn't codon bias or other types of things going on or UTRs. It really was the 5' UTR that was most important. And since he then had individual nucleotides, he could start asking the question that were causal or potentially associated, what's likely the cause here? What he was finding is that essentially the disruption or creation of untranslated open reading frames, or upstream open reading frames, were really the most important class of variant. And these were really happening in essentially thousands of genes across the maize genome, that either disruption or creation was really key and seems to be central to it. So I think we now, again, have very simple models. We can identify these potentially causal variants that would either disrupt or create new UORFs. So the last area I kind of want to talk about is how we've been working on the protein structure side of things. And this work actually got started before some of that other work, and got started in our case with Eli Rogers Melnick, comparing maize to essentially every other angiosperm that was alignable at the time. And he saw that about 10% of the genome was alignable and constrained. And that essentially, from those constrained regions of the genome, if we see segregating variants at some of those really constrained regions of those genome, those should be deleterious mutations, and very similar to what people do in human diseases. And we've tested that hypothesis in a number of different studies. And for example, from the population genetics point of view, yes, these deleterious mutations are enriched in low recombination regions of the genome. Species that undergo vegetative propagation, like cassava, seem to have an increase in these deleterious mutations. And they do seem to explain about half of phenotypic variants for yield in maize and sorghum. They weren't necessarily improving our predictions, but they appeared to explain a high level of the phenotypic variance. And so Guillaume Ramstein, through a couple different studies, was trying to see if he could really push this and improve this in the case of maize. And so he created a series, we had a large set of biparental hybrids to train on, and he could test on a diverse set of hybrids to validate those models. And what he was able to see was that models that incorporated information on where the SNPs were relative to genes had a significant improvement over standard genome-wide prediction models. But incorporating those deleterious mutations for some traits like plant height really didn't have much of an effect over just knowing where the genes were. And for grain yield, there was a modest improvement if you knew where those deleterious mutations were. But again, the scale of the improvement was modest using that type of information. And so Guillaume then asked the question, do we have potentially too much noise in these GERP estimates, these evolutionary estimates that are out there, and how can we add other additional genomic annotations? And so what he did was he essentially scored each genomic region and each protein for both protein structure, disruption scores, and so on. And then he used those as predictors to try to predict evolutionary constraint. So in the past, we've just used evolutionary constraint directly in our models. Here, he's essentially creating a random forest model to try to predict evolutionary constraint. And so he now has essentially two different ways to estimates of evolutionary constraint: the observed evolutionary constraint and a machine learning model that is incorporating key elements of protein structure and other factors of predicted evolutionary constraint. And the really interesting thing is these models were all trained outside of maize, and then he can test these hypotheses within maize. And what he sees is that we would expect that essentially rare or less common alleles with low minor allele frequencies should have more deleterious mutations, and the slope should go up. And so that's exactly what he kind of sees, is that predicted evolutionary constraint was doing a better job of identifying deleterious mutations than the observed evolutionary constraint. So adding in that information on protein structure and other factors was improving these. And so then he went back to the same types of biparental cross hybrids, trained and validated that he was working on before, and did various random prioritizations using essentially weighting those deleterious mutations. And what he sees is if he focuses in on the most extreme deleterious mutations, he's getting up to about a 5 to 10% improvement in yield prediction. And the really neat thing about this type of analysis, if you look at it from the point of view of a population, you're talking across even this diverse group of population, about 1,000 variants at the 1% tail to 100 variants in the 0.1% tail, and on the order of 165 to 15 variants per genome, which is a number that essentially probably is very reasonable for for both breeding against and for editing against. So I think this is suggesting we're getting down to a small enough group of variants that are actionable through a number of different routes. So I think we're making progress on this whole approach of how of some of these tools of being able to look at sequence and starting to understand how things are expressed, or how they may be translated, or whether or not the protein is functional. But we certainly still need to do a lot more in trying to leverage this transferability. And so I think we're at this stage, we're like everybody else using AlphaFold 2 and for as many things there. And it really does look like it's going to be a very powerful tool that we can essentially estimate protein activity just based on those protein structures. I think there's an opportunity across plants to use models that have been developed from Arabidopsis to across any of the other major plants model systems out there to start working on predicting gene regulation. I think there's enough shared physiology between plants like maize and rice that essentially what's deleterious across that spectrum is still very informative for predicting things. And I think some of the networks that we're seeing at the level of physiology, they certainly are transferable over thousands of years. And I think the area that we don't know is we'd like to be able to start using regulatory networks that are shared across the grasses to really move some of these models forward. And so I think if we kind of do that type of modeling, we're going to be able to start pulling a few key traits out of some of the species we want, so that we can really focus in on adapting maize to really these new greenhouse gas challenges. And I think some of the traits we want are things that are only 500,000 years away from the species we're working on. So maize is actually only from Zaediphyperennis is only about 100,000 years diverged. So I think if we get the right set of models, right set of predictions, we should be able to start mining some key traits out of species and moving them quickly into species like maize. So I'll end there. I've mentioned a lot of the people who contribute to the individual work I was talking about there. And just want to highlight the team is organized by a great group of group leaders, Sarah Miller, Cynthia Ramey, and Peter Bradbury. And I'd be happy to take some questions.